Our next speaker is Dr. David Furman. He is the director of the Stanford 1000 Immunosomes Project at the Stanford School of Medicine. He is associate professor at the Buck Institute for Research on Aging and chief of the Bioinformatics Core at the same institute. Previously, Dr. Furman was a consulting professor at the Stanford Institute for Immunity, Transplantation, and Infection. His work involved the use of high-bandwidth, high-throughput technologies to measure immune function in humans, as well as machine learning tools to better define the role of the immune system in disease and longevity. Dr. Furman is also the founder of 2 startup companies based in Silicon Valley, inventor of 25 patents, and has published over 30 scientific articles with over 6,500 citations in top peer-reviewed journals. With that, I'd like you to help me welcome Dr. David Furman to the stage.
Awesome. Hi, everybody. It's really exciting to present the work we've been doing over the past couple of years, really here at the Buck, and how we go from basic science to try to move this to the bedside. So I'm going to try to bring you to the moon and back. So it's really space. We're doing space biology. Let's see if this works. Yeah, a little bit about myself. I've been with Stanford for about 15 years, leading this effort. We've got another $15 to $18 million from NIH to continue the 1000 Immunomes project for the next 5 years. And as part of the effort, the Buck Institute, 3 years ago, recruited me to lead this AI platform, so research in how my own interest is really in the intersection between the immune system and how the immune system determines rates of aging. And we've published a number of papers in this area. So we have been pioneering the use of multiomics for immune system and aging. And this started in 2007 when proteomics was only 40 proteins. Now we're doing 7,000, 5,000, right? Great work, by the way, Eric. I love your work and the one that you're doing with Kristen there. So as part of this effort, Edifice Health was born. It's a company that commercializes a test for inflammaging. And it was funded by Leaps and Paladin. So I don't have to convince this audience that aging is a problem. I think if we don't solve aging, there's going to be a major disaster in the healthcare systems. So we're suffering from this as a humanity. If we don't find cures, we're doomed. And so we're spending about $3.5 trillion in annual expenditures only in chronic diseases in the US. And we're actually focusing on trying to evaluate biomarkers of aging that can ultimately lead to drug discovery and development. The big problem that the drug industry suffers from is that it's extremely slow and expensive. And you probably have seen this figure before, but 90% or more than drugs fail. And the reason is largely because we're looking at the wrong place. And I'm going to have to apologize myself to be a little bit provocative here, but most of the research that has been done in the aging space, including the hallmarks of aging, is based on worms, flies, C. elegans, and maybe mice. And things don't translate, quite honestly. So we have taken a very different approach using humans. We use human organoids, human systems, human blood, humans themselves, the entire cohorts of humans. And we believe that using this human-centered research, we will be able to translate things much more rapidly into the clinic. At the same time, the space industry is growing very rapidly. You probably have heard of SpaceX and Blue Origin and Axiom taking the lead now, and NASA being a little bit behind the scenes. But what happens is Elon Musk believes that we're going to be landing in Mars in 2024. And NASA is planning to actually land in Mars in 2030. That's real. It's happening. But this poses a huge challenge. And that is that with spaceflight, there are a number of different physiological deteriorations in different systems. And that largely resembles aging. So here's the opportunity, right? What you see in this slide is normal aging in the white lines here, and how that declines in the lifespan, and accelerated aging during spaceflight. On average, we have calculated for each one of these different physiological systems that age, that physiological age gets accelerated by 30-fold. So imagine each year you spend in an International Space Station, you would age similarly to 30 years on the ground. Now, things are not that terrible. People come back to Earth and they can live a normal life and recover for some of the physiological deteriorations that they have suffered. So we have this beautiful window where we're studying humans, healthy humans, not disease, not progeria, not Down syndrome, and we can actually develop different or discover biomarkers of aging and hopefully develop interventions to cure different diseases of aging. And I'm going to get to that in a moment. Because the space industry has shifted and now the private sector is taking over, multi-omics data is largely available for astronauts. And we're working with Axiom, we're working with SpaceX and with NASA to be able to pull all this data into our hands, combine that with ground analogs of microgravity and cosmic radiation, and be able to bring this to a new paradigm and observe aging at an altered rate. We formed the company. It's a Buck spin-off, Cosmica Biosciences, and it's based in that premise, right? And so we believe that we have now the opportunity to really develop a platform to accelerate drug discovery for longevity. So some of the team members, Chris Mason is my co-founder. He's at Cornell. Dan Weiner's here at the Buck Institute. And then there's team members, part of our team at the Buck Institute, working partly in the company. Elia Overby trained with Chris, and we have more than 150 years of combined effort and research here and expertise, both at the drug discovery and drug development side of things, also risk management, aging biology, et cetera. So how does our platform work? We have named this platform Pronto. That is because we believe we have a platform for rapid output of novel therapeutics using omics. And here's the probably the most interesting part is that we can reduce that first 4 years of drug discovery, which is part of a target validation, compound screening, and maybe lead optimization as well, by around 15-fold. And how does it work? We have, first of all, the RAMP system, rapid aging microgravity protocol. Then we do signature validation. Then we go ahead and do gene compound enrichment analysis to identify different drugs that can reverse these phenotypes. Then we test them in this RAMP system. And last, very agnostic of the disease, we map this thing to different diseases to look for indications to go after. Okay? Similar to what Eric was focusing on, we also are very focused on immune aging, cardiac aging, and neural aging. And we're using immune organoids. This is all derived from tonsillectomies. They are very functional. You toss a flu vaccine, they produce antibodies. You toss a virus, they produce T cells. Cardiac organoids are derived from iPS. And neural organoids as well. And what we do is we subject these micro organoids, and including PBMCs, to microgravity simulation. And this is a facility that we have put together here at the Buck Institute. We have 2 machines. They're called rotating well vessel that basically subtract the gravitational forces from these systems. And we can observe aging at an accelerated fashion in these tissues and in these cells. So as an example of this, what I'm showing you in this slide is the inflammatory age, so the biological age that we published in Nature Aging last year, and at 1G, so this is normal G, as everybody's suffering here from very strong gravitational forces, as compared to 0G. And you see that this increases dramatically by about 50%. We are also reactivating viruses. It's well known that humans in space reactivate a number of different herpes viruses, shingles and stuff. And we also can recapitulate that in our system. Then we have the cellular senescence Sen-Mayo score that increases dramatically after microgravity treatment. And this is probably the most exciting stuff. I'm very excited about this thing. Mechanotransduction changes completely. So this is relatively a new field, right, of mechanotransduction of aging. But what happens is under microgravity, we have a redistribution of the cytoskeleton, of the actin cytoskeleton. And the mitochondria cannot hold very well, and it becomes dysfunctional. So this thing comes before, upstream of mitochondrial dysfunction, okay? So if we could target this, this could be a really good opportunity for us to go into the mitochondrial dysfunction arena, but also into inflammaging that I mentioned, and other diseases that go with inflammaging. For example, as I'm showing you here in the top part of the slide, after 24 or 25 hours of microgravity simulation, we have a complete rearrangement of the actin cytoskeleton. There's mitochondrial dysfunction, there's less production of ATP, increased ROS, et cetera, and you have a completely dysfunctional immune system, reactivation of viruses, and inflammaging. We have a lot more data that we have now accumulated over time, and we believe that we're at least recapitulating what happens in aging for 7 of the 14 new hallmarks of aging that was published in 2022 very recently. So then we do the signature validation. This is using crew member data. As an example, I'm showing you here the ramp system and different pathways that go up and down with microgravity and inspiration 4 data. So some things go in the same direction. Most things go in the same direction here, go down and down, up and up. Some things are not completely overlapping, but the p-value of overlap is 10 to the minus 16, so it's quite significant. And last, what we do is signature validation in aging cohorts on the ground. We have access to a large number of different cohorts here are listed all in this table. So what we're trying to do is to identify targets that overlap between all these different technologies so that we can play around with compounds and be able to reverse these things in vitro and then hopefully be able to run clinical trials in space. And we're planning that with Axiom. So how do we identify the drugs? We basically use interactomes. And these are about 150,000 compounds that we pulled from the literature and that are available. And then we have a package, an R package. If you guys are interested, you're free to use it. It's called gene compound enrichment analysis for hit identification. And this is just an example here on the right hand side where you see different compounds and different genes. This includes Links 1000, Drug Bank, and also FoodDB for food compounds. And then we have the hit-to-lead process where we're actually testing these compounds in the RAMP system. And as a proof of concept, we have a compound we identified that can reverse up to 70% of the differentially expressed genes. And here, you're probably going to recognize many of these pathways that are age-associated. Erythropoietin that contracts hypoxia, PPAR alpha, and then senescence pathways, HIF1 alpha, et cetera. So the pipeline goes very similar to what Erica was showing. We're tackling immune aging, cardiovascular aging, and neuroaging. We're working with the Stem Cell Institute to get all the neural organoids, aging them faster, and then be able to see if they can reverse signatures in our RAMP system. But obviously, this will be evolving as we go and dig down into the specific diseases that we can go after. I think it's going to be more like the hypoxia, fibrosis. That's my hunch. So the longevity market is growing fast. This is part of the pitch deck. I shouldn't. Yeah, and we just opened our seed round. If you guys are interested in funding, that's my company email. But if you're interested in the science, that's my institutional academic email. And thank you for your attention. Yes, there's a question over there. Okay. Over there.
Oh, yeah. Really interesting that you guys identified mitochondrial dysfunction and it looked like disruption of mitochondrial dynamics mediated by the cytoskeleton. So at Stanford, in Daria Moshli-Rosen's lab, we discovered a whole series of peptides and small molecules that block the breakdown of mitochondria, the large number of stressors. And so I'm wondering if you guys have considered trying to target, mitochondrial dynamics and to modulate this phenotype.
I think we are very early. We need to figure out a little bit better on the causality. If the cytoskeleton and mechanotransduction is upstream, we actually need to have many more experiments to demonstrate that. So we're very early. I don't think we have considered that quite yet to dig into the mitochondria. I think more of the upstream is something more appealing to us, but we need to do a series of time series analysis that haven't been done yet, and so we're expecting to do that in the next 6 months or so. Yeah.
Thanks.
Mm-hmm.
Is this on? Yeah. So at the pharmaceuticals tend to interact with individual proteins. And at the individual protein scale, Brownian motion has far more influence than any kind of reduced acceleration. So are you looking at like a cocktail of effects that might be a more system approach? Or are you actually looking for the silver bullet to target an individual effect? And do you think it's going to work?
Yeah, at the moment we're looking at individual compounds and going one by one, but we have the opportunity now, because we're moving from a rotating well vessel to a random positioning machine, to use not just 4 wells, if you will, or 4 ports, but actually 96 well plates or even 384. So if that is the case, when we move that to the random position machine, we will be able to screen a lot more. At the moment, we're basically limited by the technology.
Oh, hi, David.
Hey.
My name is Mahmood. I'm one of the fewer non-scientists in this room. So I have to tell you, I really appreciate the top-down, how you laid the problem out. And I understand the problem, and I really appreciate the solution that you're doing. This is fantastic.
Thank you so much.
And if former VC, you've got a pitch that should work. So
Thank you so much. Appreciate it.
Okay. Thanks.
Thank you.